The overall goal of the lateral root inducible system is to synchronously induce the initiation of lateral roots in plants using hormone treatments.
Plant roots are the hidden half and are known to be extremely difficult to study. We have developed an in vitro system that allows to study the process of lateral root initiation at a cytological, histological, and molecular level.
Generally, scientists new to this method might struggle because the plants have not grown into proper contact with the medium, preventing a homogeneous induction of the lateral roots.
Visual demonstration of this method helps showing the critical steps that should be taken into account to ensure a homogeneous induction.
Although this system was originally developed in the model plant Arabidopsis, it can also be applied to other systems, such as maize, rice, or barley.
To begin, apply a 20-micron nylon mesh to NPA-supplemented growth medium using a pair of sterile tweezers. Gently push the mesh onto the growth medium using a Drygalski to make sure it is in good contact with the medium. Next, pinch a sterile toothpick into the agar to create a sticky surface on the toothpick. Use the toothpick to evenly distribute a batch of gas-sterilized seeds onto the prepared plates. Seal the plates with breathable adhesive tape and store them at 4 degrees Celsius in the dark for at least 2 days and a maximum of 1 week. This is needed for stratification and ensures synchronized and efficient germination. On the 3rd day, move the plate.